Our next speaker is Gamze from Yale University.
Hi, everybody. Thanks for being here. I'm Gamze. I'm a postdoc with Mark Gursoy at Yale University, and we work on genome privacy. And today I'm going to talk about the privacy concerns and how we can deal with them in a public data sharing mode using from functional genomics data. So I think I don't need to convince you that data is important. More specifically, biomedical data can save lives. So to have a better foundation for the healthcare system, I think it's important we need to feed the basic research we do in these areas for a better patient care. In turn, as a result, all the subjects that we use for transcriptomics, epigenetics, or genetics data increasingly becoming real individuals rather than the model organisms. And that creates a privacy problem as far as sharing goes. But we believe that data sharing is very important for reproducibility, for testing new pipelines, and for dealing with the bureaucracy that comes with the special access data. However, on the other side of the coin, there is okay, I'm missing a slide here, and I don't know why. Well, on the other side of the coin, there's the privacy concerns because sharing leaks identifying information such as the SNPs and variants of these individuals. So we can actually cross-reference these individuals. So for example, traditionally, genome privacy in the past decade focused on genetics data. It has been shown that for a long time that you can use the genetics data, the variants, to cross-reference these individuals and learn private things about them, like their addresses or their last names, or if they attended a drug abuse study. However, our lab is more interested in transcriptomics and epigenetics data because the privacy studies hasn't been studying these particular data sets in the past decade. And more importantly, I think it was overlooked because functional genomics data, the point of functional genomics data is not to identify people's individuals' SNPs or variants, but it's more like to learn biological information about diseases or different developmental stages. So functional genomics data are not tied to individuals' variants, but the data by nature of the experiment comes with a lot of variants. So I'm going to show a life cycle of a typical RNA-seq experiment, then you can understand better why we should be worrying about privacy when it comes to sharing functional genomics data. So if we want to learn about, let's say, how gene expression changes in different tissues of an individual, we can actually take different tissue samples from an individual and do RNA sequencing. And then RNA sequencing will give you these small bits of RNA sequences that overlaps with the expressed genes. So as a result, the coverage of that experiment is very different than a whole genome sequencing experiment. If you don't have an expressed gene, then you will not get any sequence from there. And then we overlap these sequences with the genes, and if you overlap more sequences with a gene, that means that gene is expressed. At the end of the day, what we are interested in to see which gene is expressed in which sample. However, it comes with the sequences of these genes that is coming from a real individual that you can actually identify variants, and in turn, as a result, you can actually see who is this individual. So the thing is, the regulations governing sharing functional genomics data is somehow ad hoc. For example, HeLa's genome is locked. You cannot access it unless you specify why you want to use HeLa's genome. But you can actually search PubMed for HeLa's ChIP-seq data or RNA-seq data and get the reads from the ChIP-seq data. So then the question becomes, these low coverage, low depth experiments, how much identifying variant they leak? Can we actually quantify it? And if there is leakage from this data, can we find ways to protect individuals' privacy while publicly sharing these data files? So we took an information theory-based approach. Basically, our analogy comes from a very noisy radio channel. So you have a radio channel where you have an input, but your signal that comes from the channel is very different than your input. And you want to know what is your input, and you want to know how much information you have in your signal. So if you're hearing a word like "the," you're not learning much about the input because it's a very common word, right? But if you're hearing a word like "Taurus," which is a rare word in our daily lives, then you actually can guess what the input might be. So we can use the same analogy. If a functional genomics experiment is a very noisy experiment where you get these noisy bits of DNA or RNA, you can actually genotype this sequencing and then get variants. And if this variant is a common variant that you can see most of the population, then you're not learning much about this individual. But if the variant is a very rare variant that you don't see in much of the population, that means this experiment actually leaks high information. So it's rather than looking at the number of variants that they leak, we look at the information content of those variants. And then what we can do actually, we can find a gold standard set, which is the whole variant set of an individual, which is available through 1,000 genomes, for example, for 2,500 individuals. And then you can look at the overlap between the cold variants and the gold standard variant, and then quantify the information content of that intersection. And that intersection will be the information quantity of that given functional genomics experiment. And then further, what we can do is we can sample sequences with the increasing amount of coverage and do the genotyping for each coverage and overlap it with the gold standard and quantify the information leakage from each coverage. And so it's not surprising that among all these functional genomics experiments, the high-depth experiments such as Hi-C gives you the most information. And also, it's not surprising that the information content increases with the increasing amount of sequencing. However, then the question becomes, if we are never reaching the whole genome, the information that we get from whole genome sequencing, is that information content enough to identify this individual? So what we can do is we can design a very simple linking attack scenario where we take a certain amount of sequencing, we do the genotyping, and then now we will quantify the information in the intersection of the cold variants and the genotypes that comes from a database. So we can do it for every single individual in the database, and then we rank these intersections, and then we find the highest ranked individual that matches to the sample that we have, and then if our highest ranked individual is far away from the rest of the population, then we can say that we can identify this individual in a database. So basically what we are showing here that all of these functional genomics experiments, even the one that is coming from single cell, for example, single cell RNA-seq, you only do the sequencing in one single cell, and you only get expressed genes in a single cell. So you can imagine how low depth this experiment is, but you can still identify the individual within a mixture of individuals if you just use the variants from those experiments. So what if the individual is not in that database? Can you actually tell anything about the individual by just looking at the variants from these low depth experiments? So what we did is we put together all the publicly available ChIP-seq and RNA-seq data, all the sequences together, and then we did genotyping on them. Here's the dark blue part. And then on top of that, we use the population linkage disequilibrium structure to impute variance. And as you can see that this publicly available data can actually return us back to the whole genome sequencing data, even though it is publicly available and this is not. And furthermore, you can actually infer some phenotypes that might be private that people may not want to disclose by using these experiments. So basically what I've shown so far that there's no question that there is leakage in raw functional genomics data. But we believe that the sharing of such data is important for reproducibility, for also doing advancement of the biomedical data science, for the rapid advancement. So we were thinking about how we can improve these data files so that we can actually release the data while protecting patients' privacy. And the good thing about this data is that we don't need the sequences necessarily. We only need to know the places where the sequences overlap in the genome. So we came up with this idea called privacy-aware binary alignment maps. Basically, a traditional BAM file, you take a traditional BAM file that you get from these experiments, and then you suppress or generalize certain identifiers in the BAM file. So basically, you remove the sequencing information, you remove any tags that will tell you that there's a variant in the BAM file, and then you create this new BAM file called PBAM. The beauty of this, we know that there are several other file formats out there, for example, that uses crypto-based approaches. But the beauty of the BAM-based privacy-aware files is that you can take the PBAM file and use it with any software that will take BAM files as an input. So the utility of these files is way higher. And also, we have a way of quantifying the utility of these files. Basically, what we do is we calculate, let's say, gene expression levels from the BAM files. And now we calculate the gene expression levels from the PBAM files. Since we removed some information from PBAM files, we actually added some noise. By looking at the difference between these reported gene expression versus the real gene expression, we can actually quantify how far away we are from the reality. Basically, it's called this epsilon value. You want the epsilon value to be as close to 0 as possible so that your PBAM file is generating the similar result as the BAM file. So I'm also happy to say that we are now a part of GA4GH file format task team. So we are working on standardizing PBAM files for use for everyone. So what we did is to show the utility of these files, we calculated the gene expression levels by removing certain variants from the data. We removed mismatches, we removed indels, we removed structural variants, we removed everything. And for each case, we calculated the results from the PBAM versus BAM file and calculated the epsilon. As you can see that our epsilon values are very, very close to 0, so that the utility of these files are high. So these PBAM files works really well with certain functional genomics pipelines like STAR, RSAM, MAX2, and we are working on to see other pipelines are working as well with PBAMs. And also, another beauty of this file format system is that when we have a PBAM, we also add all the private information to a small file called .diff. So that's the difference between a BAM file and a PBAM file. But we don't put any sequencing information there because you can get the sequence from reference genome already. So the files that you want to store safely and move is very small in that case. You don't need to store BAM files. You can just store the .diff files and lock them up. And we have these software suite called ptools where you can take a BAM file, convert it into PBAM and diff file, and then you can also go back from diff and PBAM using the reference genome that was used for that particular experiment and go back to the BAM file without storing the BAM file. And we are also working on developing the ptools to work with CRAM files as well. So combined with this work and our previous work done in the lab that was also supported by BD2K, we can now actually quantify private information leakage at every step of functional genomics data summarization. So what I showed you is here where we actually see the leakage from the raw reads and the BAM files. We actually showed this year that we can quantify the leakage from the signal profiles, and then in 2016 we showed that we can actually quantify the leakage from the gene expression matrices. So as you can see, our modified reads where the PBAM files sits here. So in this triangle, as you go down in the triangle, the utility of these files are huge because they are raw files. You can do anything with them. So our PBAM sits in a very good place where the utility and the privacy is very balanced. So with this, I'd like to thank Gursoy Lab, members of Gursoy Lab, specifically Mark, Arif, Molly, and Fabio for helping me with this. Our collaborator from Berkeley, Professor Steven Brenner, BD2K Young Investigator Award, and ISMB Travel Award for bringing me here. Our lab is hiring postdocs. So if you're interested in genome privacy or biomedical data science, please apply or come see me. I can talk to you on behalf of Mark. And also, we are right now chairing a session, a biological data privacy session at PSB this year. If you have work that you can't wait to show us, please come to Hawaii with us. And I also would like to thank all the HapMap and 1000 Genome individuals for giving their biosamples to the science. It didn't only help us for biomedical data science, but also help us to see if there are privacy leakages in certain data files. And thanks everyone for listening.
I have a question. So since the p-band file only contains sequence from the reference genome, so why don't you use BAM file instead of BAM file?
Which file, excuse me?
The p-band file. Why don't you use p-band file?
P-cram file?
Because you only have reference genome.
Yeah, you're right. Well, we are working on that. So it was the first step was the easier step is to deal with BAM files. But now we're working on using the CRAM files, which will make all the other files a lot smaller than the current ones.
I'm just wondering how easy it is to actually recover the individual identifier from the BAM files. Like how well can it predict accuracy of that person segment?
That's a great question. Well, all our experiments are done on one individual, NA12878, and with that one individual, even from very, very small BAM files, we were able to always identify from the 1000 genome panel. But now we are working on adding like 10 more individuals so that we can actually calculate the accuracy. But to be able to calculate the accuracy, we have to have more individuals. And unfortunately, there are not many individuals that the functional genomics was done, because this is a new area now that we are doing functional genomics to real individuals. So far, I can't tell.
Well, I guess the question is like, do you think I guess like thinking back on that, do you think in the future, because there are more individuals and more in the cohort, it will be harder to actually predict the individual from the
We think it's the opposite. So for example, what I did is I simulated a lot more individuals and put it in the database. So what does it do is it makes certain variants even more rare. So if you have, let's say, a variant that is in the 30% of the population, if you add more people, you sometimes end up having that variant only become in 10% of the population. Then it makes it easier to identify this person. But again, all these experiments are done on NA12878, so we need to do more experiments on different populations. Like, for example, Africans have different range of variants than the European population, so it might be a different case for them.